Hello, and thank you for coming to my virtual ESA talk. I'm Tad Dallas, an assistant professor at Louisiana State University. But here I'll be presenting on some work that I did as a visiting postdoctoral researcher with Pedro Hordano. I was working at University of Helsinki at the time on some applications of joint species distribution modeling and some spatial synchrony work, and I thought of the potential that joint species distribution models could have for understanding host-parasite interaction networks. So I pitched this idea to the HPC Europa 3 folks, which is a great funding opportunity, trying to give researchers access to high-performance computing systems for research exchanges. I was lucky enough to receive the funding, but the data actually sort of weren't there for what I pitched. And so I pivoted to a bunch of different projects, and this is one of them. This is a very easy pivot to do in Pedro's lab, which was just a really warm and intellectually curious place, as Doñana Biological Station in general was. And one of these ideas that we developed during this time of pivoting was to apply some of the same ideas that had been applied to species area relationships to ecological networks. And so species area relationships basically say, at least the mainland species area relationship, which is what I focus on, increasing geographic area means you find more species. The idea here is that it also could influence species interaction networks. And by species interaction networks, I mean the associations between host and parasite species, but not with sort of like host-host interactions through competition and other things like that. This is sort of pictured in the slide, taking work from what I was doing at University of Helsinki around this time. And I guess that was 19 or so. Where this interaction network is between a bunch of small mammal hosts found in a desert sampling for part of the Speciesarea long-term ecological research project, and their interactions with a sort of collection of endo and ectoparasites, where these colored boxes correspond to known interactions, known associations between hosts and parasites. And these interactions sort of form a network that we can examine, not only through like local properties like the number of interactions per host or something like that, but also through structural properties of the entire network. But that last network, the Speciesarea LTER network data, is a single location in space. And for that, we can't examine mainland species area relationships with a single spatial sort of data point. We need sort of broadly distributed data. And if we had that, if we had multiple networks across space, like pictured here at the global scale, where we have information on the log number of species interacting or species present at a given habitat, this would create a system where we actually had, at each one of these colored points, a network of host-parasite interactions. And this would be a sort of absolute necessity to look at species area relationships and to look at network area relationships, which is what I sort of was really focused on. And so based on previous research, we sort of know we definitely know how species area relationships should look, right? And so that's the top left panel where we have, with increasing cumulative area on the x-axis, we have species richness increasing rapidly and then saturating. And it saturates because eventually you're going to sample the full contiguous area that you have. So like in the case of the previous slide, it'd be like the entire globe. You know every species that is on the globe. And you slow down because like you start sampling species you've already seen before. So it's more of a sampling process. And we also have ideas about how this would affect network structure and different aspects of networks. And that's mostly due, not mostly, it is due in some part by some of the really great work led by Nuria Galeana. So that paper from 2018, it's cited at the bottom. If you're interested in these ideas, that's a starting point. Like, read that before you read like the paper that this is based off of. And so in the top right panel, the authors are sort of going over what are the expectations, not only about just species richness, but about the actual structure of species interactions with increasing area, where we might expect that the average number of associations per species in the network, as in the panel B there, will do the same thing as species richness. It'll sort of go up and then saturate until a point where most associations are known, and you're just sort of resampling. And so the actual number of links per species is harder to gain later on in the sampling process as you increase in area. But one thing that struck me was this idea of overall network structure changing. And so in this paper, they suggest that the overall structure of the network, measured as modularity in the bottom right panel, wouldn't really change with increasing area. So that is, like, the network will not become more sort of compartmentalized. I'll define modularity a little bit better later on, probably. But the network won't become more compartmentalized with increasing area, with the caveat they set up being that in an incredibly heterogeneous and sort of segregated habitat, you might imagine that modularity would actually increase as a function of area. So it's either not going to change or it's going to increase, right? So we have these like set of hypotheses about how we might expect species area and network area relationships to look. And thanks to some really hard work from the folks over at the London Natural History Museum, we have a global data set of host parasite, specifically host-helminth parasite associations, geo-referenced to geopolitical boundaries, which I fully recognize, not ideal, but it's global. These data are fantastic and one of the largest sources of host parasite association data that we have to date. And so coming from over 2,800 published studies, over around 250,000 interactions, fantastic data. Sort of seconded only by some of the more recent work I've been involved in with the Virion consortium. So I cannot plug them enough. Go to www.viralemergence.org for more information on that. And the Virion data set is fantastic. But let's stick to helminths for now, because helminths are awesome. In this map that I showed earlier, it was sort of a teaser, because this is the actual distribution of those data. And so for all of these geopolitical units, which correspond to sort of states in the United States, countries elsewhere, we have information not only on the geographic area of that sort of encompassed by the arbitrarily drawn lines on the map, but we also have its known host-helminth interactions. And so we can form networks for each one of these points and test network area relationships. And to do this, we took our set of networks and calculated the distances between all of the geopolitical units. Then, starting with 50 randomly selected geopolitical units, we constructed the network for that given unit, that location, and iteratively added the nearest neighboring unit to the network. So building up that network, globbing on networks to networks. And at each iterative step, we counted the number of host species, the number of helminth species, the cumulative area, and measured modularity and other network process. This additive process eventually meant that all of the networks grow in size until the cumulative geographic area is the full network, the global network of all host helminth associations known. And so to go back over our hypotheses, we say species area relationships will exist. So both helminth and host species relationships or richness will increase with increasing cumulative area. There's an idea that helminth richness should increase faster. If you want more information on that, check out the paper. I'm not going to go into it. I had the idea that connectance, which is the fraction of realized interactions in network, would do something. And there's also a ton of sort of theoretical reasons why it'll do something, and I don't have time to go into it here. But check out the paper or the Galliano paper. And then finally, we thought modularity, we measured it, but we're sort of like, let's see what happens, but it's predicted to do nothing, right? And what we found was positive species area relationships generally in the mainland case for both hosts and helminth parasites, positive relationship for number of links per species, neither of which were sort of saturating, both on sort of a log-log scale. We also saw that connectants declined precipitously as you increased log cumulative area. Finally, we looked at the modularity, which was either suggested to like not change or increase over time, increase over cumulative area, and it didn't. It actually decreased. And so this is a muddy relationship, but it is a significant decrease, suggesting that with increasing area, globbing on networks, they tend to become less modular when standardized for like things like connectants and other things that might influence modularity. These are the findings recapitulated here. A quick plug. I'm looking for opportunities for graduate students and postdocs. I'm currently at LSU, Louisiana State University, but we'll be moving to University of South Carolina in the spring of 2022. Go Gamecocks, I guess. And finally, I would like to acknowledge several funding sources that gave me the privilege to do this work. Sevilla is amazing. My Spanish is terrible, but I enjoyed every minute of the experience. Running along the river, drinking coffee in the plaza, trying to wait to eat dinner until it was acceptable by Spanish standards. All of it fantastic. Special thanks to my collaborator on this, Pedro Hordano, his entire lab, and Doniana. Amazing folks. And thank you for your time.